first stage to mitosis. During the prophase, the DNA in the cell coils up and becomes visible as chromosomes. Each chromosome has two identical parts called sister chromatids. The nucleus slowly begins to break down so the chromosome can move around the cell. While that is happening, spindle fibers start to form and move to the opposite side of the cells. By the end of the prophase, the cell is ready for the next stage of the metaphase.
Metaphase is the second stage of mitosis, the process cells use to divide. By the time a cell reaches metaphase, the chromosomes have already condensed and became clearly visible. During metaphase, all the chromosomes line up across the center of the cell along an imaginary line called the metaphase plate. Each chromosome is made of two identical sister chromatids, which are held together at a region called the centromere.
Spindle fibers, which formed earlier, attach to the centromeres of each chromosome. From opposite ends of the cell, the attachment is important because it ensures that when the cell divides, each new cell will receive an extra copy of every chromosome. Metaphase acts as a checkpoint for the cell. The cell does not move on to the next stage until all chromosomes are properly aligned and securely attached. Once everything is in place, the cell is ready to move into anaphase, where the sister chromatids will be.
During anaphase, one of the most important stages of mitosis.
During anaphase, the complicated chromosomes are finally pulled apart. Each chromosome, which was made of two identical sister chromatids, splits at the centromere. When separated, the sister chromatids are considered individual chromosomes.
Spindle fibers attached to the centromeres begin to shorten. This action pulls the chromosomes into opposite directions, one set toward each other pole of the cell at the same time. The cell starts to stretch and elongate, preparing to divide.
Anaphase is crucial because it ensures that each new cell will receive an identical and complete set of chromosomes. If this step does not occur correctly, cells may end up with too many or too few chromosomes.
By the end of the anaphase, the chromosomes are clearly separated and positioned at opposite ends of the cell, setting the stage for the next phase, telophase.
During metaphase, all the chromosomes.
The video has already stopped.
Telophase is the final stage of mitosis when the cell begins to finish dividing its nucleus.
During this stage, the chromosomes that were pulled apart in anaphase reach opposite ends of the cell. Once they arrive, the chromosomes start to uncoil and change back into a less condensed form called chromatin.
Next, a new nuclear membrane forms around each set of chromosomes, creating two separate nuclei within the same cell. The nucleolus reappears inside each nucleus.
At the same time, the spindle fibers that move the chromosomes break down and disappear because they are no longer needed.
Cytokinesis is the final stage of cell division, and it begins right after the nucleus has finished dividing during mitosis or meiosis. While the genetic material has already been separated, cytokinesis is a process that physically splits the cell into two independent daughter cells.
In animal cells, cytokinesis occurs through the formation of a cleavage furrow. A ring of protein fibers forms just beneath the cell membrane at the center of the cell. This ring tightens and contracts, gradually pinching the cell in half, much like pulling a drawstring. Eventually, the membrane closes completely, placing two separate daughter cells, each with its own nucleus and cytoplasm.
Cytokinesis is essential because it ensures that each new cell has the proper structures and materials needed to function. Once cytokinesis is complete, the cell cycle can begin again, allowing for growth, repair, and reproduction in living organisms.